everyone, good evening and welcome to yet another Eminent Scientist lecture. And today I'm very happy to share with you that we have with us our eminent speaker, Dr. Anshu Bhardwaj, who is a senior scientist from one of our CSIR sister laboratories, that is Institute of Microbial Technology from Chandigarh. A young scientist and also who has been recently recognized as one of the young innovators from India by United Nations Development Program. On behalf of my director, Dr. G. Narahari Sastry, and the entire SRTP team here at CSIR NIST, I welcome you, ma'am, and I thank you for accepting our request to deliver today's lecture. We're very happy to have you with us this evening. And I would like to share with you that your lecture, we are live on MS Teams, Facebook, and YouTube. So before I request you to start your lecture, I would like to request my young friend, one of my young friends, Ms. Monty Gogoi, to give a brief introduction. And then after that, I request you to start your lecture. Ms. Monty.
Thank you so much, ma'am. Dr. Anshu Bhardwaj is a biologist and data scientist. Presently, she is working as senior scientist in CSIR Institute of Microbial Technology, Chandigarh, and as assistant professor, AcSIR, Delhi. She is also a long-term fellow of CRI, Paris, France, and is also team lead Drug Discovery Virtual Tool Room Drug Discovery Hackathon 2020, Government of India. Dr. Anshu Bhardwaj obtained her PhD in 2008 in understanding genotype to phenotype correlations in human mitochondrial diseases from CCMB, Hyderabad. She then joined CSIR as one of the founding PIs of the open source drug discovery project in 2008. She conceived, designed, and implemented crowdsourcing as a tool to tackle challenging scientific problems, a futuristic approach to drive biomedical big data projects, and lead to human resource development in frontier areas of data science and analytics. She has over 40 publications to her credit and has trained over 1,000 young students. She served as an associate scientific advisor to Science Translational Medicine and is on the editorial board of Frontiers in System Biology and Journal of Genetics. She has several honors and awards to her credit. She was one of the 4 young innovators in India, United National Development Program, selected for the prestigious International Visitor Leadership Program by US State Department, awarded Newton Bhabha Fund from the British Council and the Royal Society of Chemistry, UK. More recently, she was also selected as the first female long-term CRI Fellow, Paris, France. Her passion is biomedical big data analytics with focus on understanding infection and rare disease biology towards better therapeutics. She has conducted several workshops, has been invited to deliver about 150 talks at national and international platforms. She has immense experience in handling open interdisciplinary communities for large-scale drug discovery projects. Now I request ma'am to please give your presentation. Over to you, ma'am.
Yeah, thank you so much. And at the outset, I would like to really thank Dr. Shastri for inviting me and giving me this opportunity to speak with all of you. So, can you confirm if my screen and slides are visible?
Yes, they are. Just we are sharing the slides. I think you can see now.
All right. So a very warm welcome to everyone who is listening to this presentation. As you can see, the title of my talk says Believe, Learn, Share, Experiences with Open Structured Collaborations. As I move forward in the presentation, each of these words will start making sense to you. And we'll consolidate what we have understood towards the end of the presentation. So putting in context and scope of where do you need to collaborate and why, and what are the challenges we are dealing with, I'd like to start by actually introducing you to the concept of antibiotic resistance. Now, as you can see in the graph here, there are actually already existing subset of bacteria that are resistant to antibiotics. Now, all of us, when we get sick, when we have infections, particularly bacterial infections, how do you treat them? You actually prescribe antibiotics. Now, these antibiotics end up killing the susceptible bacterial population. And if a patient has all susceptible bacteria, the patient gets cured and there is no problem. The problem happens when one of the bacteria in the population is a mutant and the antibiotic does not work on it. So as you can see in the middle part of the slide, a single bacteria that was resistant survived the antibiotic attack. Rest all the susceptible ones died. And now this single resistant bacteria actually grow and proliferate further. And now in order to treat this bacteria, we know that this antibiotic will not work. So we will have to end up discovering new antibiotics. Now this is a very static picture. How do we see things in real time and understand what is happening around? So a couple of researchers from Harvard did a very interesting experiment a couple of years ago. What they did is they actually showed everybody in the world how you can actually see drug resistance emerging in real time. So what they did, they actually, as you can see on the slide, they made a huge petri dish. Now this petri dish is like 4 feet and 2 feet, actually the size of your dining table. It's a huge petri dish, and they poured media, food for bacteria, that is laid on it with some ink so that the media looks black. And when bacteria grows on this media, they will appear white. So with that contrast, you can visualize the growth of the bacteria. Now what they did, they divided this particular big petri dish into multiple channels, and with each channel, they actually increased the concentration of antibiotics. Like say, for example, at the far end, there was no antibiotic, and then they started increasing the concentration of antibiotic 1 to 10 to 100 to 1000. And this is in context of the fact that this range 1 was basically just enough where the antibiotic was able to kill the organism. So you know that it will kill the bacteria at 1 and it won't grow till here. What they did is then they poured E. coli at these 0 concentrations and let these bacteria grow. What they noticed is what you see on your right. Now, as I told you, since the ink was black, E. coli was appearing white on this media. And if you see, the E. coli actually kept growing into higher concentrations of antibiotics. So from 0 antibiotic till almost 1000 times the concentration of antibiotics, these bacteria grew. So the observation is that bacteria actually spread as they evolved. So they actually, the bacteria got selected for a specific mutation that is making them resistant to this antibiotic, and then they keep growing as the antibiotic concentration kept on increasing. There is a very nice video around this experiment, and that can be searched on the internet with the mega plate. Now what this tells us is that, and is the fact that all of this can happen in a very short span of time. So in this experiment, they observed that bacteria move from 0 concentration of antibiotic to a 1000 times concentration in a matter of 10 to 11 days. So that is how the drug resistance appears. Now, is it only in vitro, or do we really see this phenomenon in real life? So this is basically a timeline. It's a very busy slide. Don't get confused by looking at so many things. The objective is to show you that if you look at the top panel, it shows you when was an antibiotic introduced. The bottom panel shows you when was the first time the resistance to that antibiotic was reported. Now, if you look at examples like levofloxacin, you see that the same year the antibiotic was introduced to the patient, it already was showing resistance in some part of the population. Now, this actually indicates to us a very clear phenomenon. Antibiotic resistance is an evolutionary phenomenon. It will happen, and that is how bacteria has survived on this planet for more than 3 billion years. And all of this evolution has helped them to learn better when you actually insult them with newer and newer antibiotics. The problem actually intensifies, and with the fact that now these antibiotics, which are supposed to be used only by human beings, that too in case of infection, are being used rampantly in several other scenarios. So there is a huge antibiotic misuse. Say, for example, we actually use colistin, which is one of the last line antibiotics, as a growth promoter in poultry. And since bacteria already see these antibiotics, the ones that are already resistant get selected for, and you don't see a susceptible population at all, as we saw in the first slide. So the point is, we are dealing with a situation where we need to come up with new solutions and new antibiotics faster, or we need to identify or control these infections. Now, there are several ways in which infections can be dealt with. You can make very accurate diagnostics, so you don't prescribe antibiotics either when they are not needed, like say, for example, when you have a cold and flu, which is mostly viral in nature, or you have more targeted diagnostics so that a gram-positive infection is treated by an antibiotic which is focused on gram-positive or not the other gram-negative ones. You can also have vaccines to prevent infections altogether, or you can actually have much more public healthcare intervention if you know the disease epidemiology better. But today's focus of my presentation will not be diagnostic vaccines or epidemiology, but we'll be focusing on drug discovery. We'll understand what goes into drug discovery of a new antibiotic, what I have learned in the process, and how that learning can help us grow and do better science. Now, there are certain terms that we need to be clear when we are talking about drug discovery. When we take, all of you might have taken antibiotic at some point in your life. It was either like a capsule or a tablet. And when you take it, you feel better. What exactly goes on there? So this drug actually interacts with a protein in the pathogen in your body, which is responsible for the pathogen survival. So by definition, a drug target, something that is targeted by the drug, is a molecule in the organism or an agent, which is usually a protein, is intrinsically associated with a particular disease process and produces desired therapeutic effect, which means you get cured. And drug simply means that it's a chemical or a biological substance like antibodies that can have some kind of physiological biochemical effect on our bodies, and we get treated because of that. Now, one thing that one has to remember very carefully in this situation is that identifying a good drug target or getting onto a drug, neither of them is an easy process, and I'll tell you why. Now, selectivity is very, very critical. I'm not sure how many of you have played this game, but the point is, if you assume that the arrow here is the drug and this apple is the target in your bacterial system, then imagine what happens if the arrow misses. Either the drug will not be effective or it will end up hurting the human host. So what you need to do is to design drug molecules which are very specific to the target, and we'll see an example of how do we do that. Now, selectivity is just one property of a good drug. Properties of an ideal drug include many, many things, like it should be specific, it should be efficacious. When we say efficacious, what it means? You cannot take 10 or 12 capsules in a day. So it should be able to work in very small amounts. And of course, it should be non-toxic. It should not produce effects that make you feel bad. And in order for doing that, a good drug should be absorbed very well in our body. It should be distributed across different tissue systems so that the site of action can be reached. It should be metabolized because it's a drug, it's not food. So you need to metabolize and excrete it out of the system. So you should not let the drug accumulate. So the point is, a particular drug has to have all these properties to maintain the therapeutic effect as shown in the graph here. And balancing these properties is a very, very hard job. Now the point is, you may ask me, oh, all right, we have been talking about drugs and so many properties. What drives these properties? So a single factor that drives these properties is the structure of these compounds. Now, as you can see here, I have shown you an example of an anti-diabetic drug. Now, the top one here actually is very efficacious, but then it shows bone marrow toxicity, so you cannot use it. Now, what you do is you replace one small functional group and a toxic drug becomes a safe drug. Now, you pointed the take-home message from this slide is that structure of a molecule derives its activity. So if you change the structure, the activity should change. And if you understand which part of the structure change leads to what kind of activity, you can develop a molecule which is not just specific, but it is also efficacious, it is also non-toxic. But at the end of the day, understanding of this chemical structure is very, very important. Now, what happens if you don't understand this very well? I'll give you a very standard example, a very textbook example. This is a drug which is called as thalidomide. It was prescribed to pregnant women early in 1950s and 60s to basically combat morning sickness so that they can sleep well, right? What happened and what they realized after a couple of years is that almost 10,000 kids that were born to mothers who were actually taking this drug, thalidomide, during their pregnancy, end up giving birth to kids with deformities. Like, say, for example, one of the major deformity observed was phocomelia, in which very short or absent long bones and flipper-like hands and legs appear. So these kids actually don't have appropriate body parts. So you can look at the adversity or the impact of a molecule which is showing the desired effect. So the pregnant ladies were not feeling the morning sickness, but in turn, the kids who were born from these women actually were malformed. So one needs to be very, very critical in trying to understand what is toxic and what is not, because it can have a long-lasting impact. Now this, we understand that we need to find something with several properties at the same time, and that is why drug discovery is a complex, risky process with high iteration rate, because finding all the properties in one molecule is extremely difficult. And at the end of the day, when you start with several thousand compounds, you may end up with one drug if everything goes fine. And as you can see here, I have listed clinical trial phases. Given that we had such examples in the past, safety in humans is one of the first things that is actually tested in clinical trials right now. If it is not safe, it is not taken downstream. You can also realize that the amount of time that I have mentioned here, it takes a very long amount of time to bring a drug from discovery to something a patient can use. And this also tells us that it's a hard, long-drawn process. It needs several people. Like, say, for example, if I'm working on an antibacterial, a microbiologist will have to first grow the bacteria for me to test a compound. For me to test a compound, a chemist first has to synthesize it. For a chemist to synthesize, somebody has to design it. So a medicinal chemist will do the design. So you can see a medicinal chemist, actual organic synthetic chemist, a microbiologist. Then you actually come with what the host does to the drug, which is your pharmacokinetics, and you need the NPK experts, and you need to do mice model studies, you need to do human clinical trials. And right now, in the era of a big, data-driven scientific discovery, you also have people from systems biology, chemoinformatics, and a very large community of people who can do a lot of data analysis before some things can be taken downstream. The point is, the kind of infrastructure, the kind of facilities, and the kind of skills one needs to drive a drug discovery process is very, very varied. And that's why even big pharmaceutical companies actually have their offices and their sites distributed across the world. It's very hard to build in all the expertise under one roof. But the problem is, when a molecule in a company fails, in whatever stage it fails, nobody knows what really went wrong. So the whole point was, if we don't know why we are failing, how do we find ways of improving and succeeding? So instead of a conventional drug discovery model, which I have shown here, why not use an open innovation approach? Just unlock. Let every research group or with industry collaboration, individuals participating, look at where we are failing and if there is already something that they can help with. It also will allow others to know that these routes actually lead to failure. So reinvention of failures will not happen. So everybody will save time and resources and can arrive at solutions faster. And this can happen only when you openly share data for a wider community to use. This is what was done as part of the open source drug discovery model, where a large number of PIs, as you can see marked by different colored circles, each color expected to be a different skill. And when they came up with different set of ideas, a team emerged. Because a team, you need somebody who can work on the assay system to test the compound. Somebody can synthesize the compound, the others can design the molecule. So the point is these teams need to be formed, which is very, very unconventional. It may sound very regular, but it is not. It's a very unconventional setup of teams driving projects into translation as a drug discovery pipeline. But as you see, the funnel is porous, which means that every data point, every idea, every method is open for anybody in the world to use. And I'll come to the structured collaboration part. When we are working in such an open, chaotic system, there has to be some structures in place for us to not get lost. So that will come a little later. Now, how do you achieve this? So there is one concept which is called as crowdsourcing, which means that how do you harness the knowledge of the crowd to achieve common goals? And if you can see, the term was coined only in 2006, but there are several examples of crowdsourcing dating back to like 200 or 300 years old. The basic principle of crowdsourcing is you have a big problem, in our case drug discovery, we know how difficult it can be. You divide that big problem into smaller tasks with small individual contributions, and it becomes a larger goal. Just imagine it like this. I gave you a burger which is this much in height. You cannot bite it, right? But if I give you small burgers, everybody can bite and eat. So you need to translate a bigger problem into smaller pieces and achieve larger goals. And existing technology actually makes it much more exciting and feasible. So where did we apply these concepts? So we applied these concepts to tuberculosis for a very simple reason. India is the hardest hit country in the world when it comes to TB. It's the top infectious killer in the world today. And if you see, it is not just the impact in terms of human life, it also has impact in terms of economically, it actually has a huge economic burden on the country. So the people are suffering, kids cannot go to school, and it actually was a very obvious choice to pick tuberculosis for us as the first target disease. And with TB, the problem is this. So the problem for drug resistance in TB is its long duration. Now, if you look at the graph on your left-hand side, what it shows you on the x-axis is time in months. On the y-axis, it shows you bacterial load in sputum, in what you cough out. And what you see here, that a typical treatment duration for a drug-sensitive TB, it is not yet resistant, so you know that the drugs can really kill all of the bacteria, is actually 6 months, right? For drug-resistant TB, where we know that some of these drugs have gone non-functional, and that is why you need new drugs, the treatment duration is even longer, as long as up to 24 months. A typical multiple drug-resistant TB patient end up taking approximately 15,000 pills during the course of the treatment. It is unthinkable. The treatment is more difficult than the disease. So the problem is, how do you deal with such a difficult situation? Why it takes TB 6 months of treatment for drug-sensitive TB and 24 months for drug-resistant TB, and when you talk of XDR, it's almost fatal. So the point is, probably we don't understand the TB bacterium so well to be able to actually impact it at the right place. So that is when I am sure many of you have seen this graph before. So these are 6 blind men actually trying to look at an elephant. And if you see, none of them can actually imagine it to be an elephant. Somebody is assuming the elephant's tail is a rope. Somebody is assuming that its trunk is a snake. Somebody is saying the ear are fans, but none of them is saying it's really an elephant. And that's the problem. Till the time we holistically understand the problem we are dealing with, this will be our interpretation, and it's not going to take us anywhere. The same thing applied to big data, the era of big data. We are still blind men. We say, okay, work on protein X. I think it is very important for TB survival. Somebody says, no, I think compound Y should work better. Or, some compounds in combination can work better. But are we talking in a holistic sense here? Or are we still doing the same thing of picking up a piecemeal approach and not leading to anything conclusive? I like to give you a simpler example for this. I'm sure most of you have watched this movie. Yes, it's the Polar Express. I'm sure all of you have watched this. It's the Avatar. Why I am showing you movies in a scientific talk? Now, these movies were made on a very new concept, which is almost 20 years old, of synthespians or digital doppelgangers. How do you digitize actors so that you can control their emotions? So you will not need an actual actor to act in movies. They will look like natural actors, but they won't be. So if you really see, both Polar Express and Avatar are not categorized as animated movies. You actually can see real people in this picture. So the point is that even the entertainment industry is looking at development of these technology, where if you look at how these movies are made, the actual stars are not doing any actions in the movie. So, but their expressions are captured. How? There are a lot of sensors which are placed on their face, and when they act, these sensors transmit the emotion and placed on their digital version. These are sometimes also called your ghostly twin. The point is, the take-home message is, what if we can generate digital avatars for pathogens? And this is nothing but in technical terms is called computational models of organisms. In our case, because we are dealing with MGB, it is going to be a computational model of MGB. It's very simple. If I have to simulate something in a digital form, like myself in digital form, I should first need to document who am I, right? Likewise, if I need to document a pathogen in a digital form, I need to know who the pathogen is. And that is when you actually need to identify all the parts, all proteins, all metabolites, all macromolecules in the system. How do they interact with each other so that I know the emotion, and how these interactions are regulated in different conditions. When the actor is meeting a friend, they are happy, but they are meeting their enemy, they are either sad or angry, right? So you know the emotion conveys the context. And can these help us in understanding how resistance arises in the very first place, so that we can identify ways of dealing with it in a computational sense even before setting up experiments? Now, how do you connect the dots? It's a pretty hard job. So if you go, for people who are new to the scientific literature, a lot of it is available from PubMed and NCBI. If you go there and do a simple search on Mycobacterium tuberculosis, you will realize that a couple of years ago, there were around 800 papers per year. But as of now, there are more than 3,000 publications on MTB per year. So even for somebody working in the field, it's not possible to follow all the work that is being done in the world, which means that now one person's intelligence is not enough. And that is when you need to bring a larger community in a crowdsourcing fashion to actually make us understand how to basically simplify and understand the complexity of these organisms. So of course, our agenda was, how do we actually understand what all has been done, that all research that has been done on MTB till date, convert that into something that actually allows us to understand the organism, right? Giving you again an example, how do we actually connect the dots here? Now this is a Google map. Some of you who are in Delhi or you have been to Delhi at some point, you know what I'm talking about. It's the Rajiv Chowk or the Connaught Place as it is more famous. And Connaught Place is typically famous because it has several entry-exit points, right? Now the point is, if I need to capture somebody in the Central Park and not let them leave from any exit points, I need to map all the exit points and close them. If there is any exit point I have not mapped, this person can actually escape from here. Now this typically happens when you develop a drug and the organism develops resistance against it. It identifies a gap and escapes. Now the point is, for me to understand, okay, how do I map or how do I block all these paths? I first of all need to know what paths exist. Translating the Google equivalent map into an organism context, it actually equates to the fact that how the information flows in an MTB cell what are the kind of metabolic reactions are going on, and once we have mapped this, then we can identify separate ways of choking the system so that there is no bypass, the system cannot escape, and there will be no drug resistance. Of course, it's pretty hard because it's an evolutionary process, but we can at least make a more informed decision in prioritizing these candidates. So what we did a project which we called as Connect to Decode, and a lot of experimental studies were buried in literature. Everything was manually mined, and all of this curation was done with the help of volunteers who joined the project, and we generated a very comprehensive protein-protein interaction and metabolic map of TB. Okay. Now, all of this work was done online, and we generated, so basically, in addition to generating the interactome and metabolic map, we also understood the functions of 87% of what MTB genome can do. So we actually improved annotations. Now, this is fun because when we were to close the annotations and refine them, we went from online curation to on-site curation. So all the top contributors online were invited for an on-site curation exercise for a couple of days. And when we were doing this curation, believe me, it's a very tedious and hard job. You have to read papers systematically, one after the other, trying to say, okay, I understand the function of this protein interacts with some other protein, and start putting that data in a very structured fashion. It's a very hard job. So when we were doing it as a mundane activity throughout the day, the productivity was very, very poor. So we followed a 90-10 rule. We actually had power play sessions. So every alternate hour, we just used to sit and do annotations. The next 1 hour, you're free to go and play. So we played. Then again come back, 1 hour to do the annotations, the next hour take a break. And the minute we started doing that, those spurts of focus every alternate hour improved our efficiency multiple folds. So this was one interesting thing when you learn and work with the community of what new things you can devise so that the activity becomes playful, enjoyable, and everybody learns in the process. And this is basically again to show you that when we launched the map, some of the world's top leaders on systems biology joined us on an on-site open collaboration, and with so many people working together and trying to solve the problem. So this activity got the whole world together looking at TB and to see how we can understand it better. Now, this is the map that you see in the inset here, so dense, is what we actually generated. 5% of this map I have wrote it on the rest of the slide, so you know how complex data was generated from this exercise. Now, you will ask me a question, okay, I see a lot of nodes, which are your green and red and purple boxes. I see a lot of lines connecting them. What am I supposed to do with this? Like, what does it really mean? So this actually tells me all the routes in MTB. Now, how do you identify routes that I block and MTB dies? It's not very easy, at least visually it looks extremely difficult. That is when we take help from graph theory. And there are several measures of centrality in graph theory. The ones that we have used in our case was betweenness. Now, if you look at the bottom of the slide, in very simple terms, what betweenness centrality does is that if you see these are betweenness centrality values of each node in the network, the ones with the highest betweenness tells me that if I remove this node, which is number 4, which it is assumed each node is equivalent to a protein in MTB. If I remove the 4th node, then 1, 2, 3 graph gets separated from the rest of the network, which means there is no bypass route by which this part of the network can see the remaining part of the network. So practically, I have divided MTB's information flow into 2 separate halves. That means it cannot survive. So when you curate this information, generate a network, this is what we did, and we calculated betweenness. So higher network importance means higher betweenness. So when we applied this concept to the entire set of nodes in this network, we came up with 73 such central proteins that when removed were actually breaking this big network into 2 different pieces, which means a lot of information flow between MTB was already happened. But now don't forget, these are important proteins for mycobacteria, right? But when you want something that is also specific to MTB, specificity. So we had to eliminate all other proteins that were also common in the human host. This is a very important point. And when we did that, we came down from 73 to 17 MTB drug targets. I will not be going into details of what each one of them does, but the take-home message is that we actually did a very large exercise of crowdsourcing, generated several networks on MTB, applied several mathematical models, eliminated anything that belonged to the human host, and identified targets for TB. Now, target identification, actually the next obvious thing is now to publish it. So now you can see that a large number of these authors that you see here have contributed in development of these platforms, and they are part of these major publications. The world noticed what we did, and we have been cited in several places as the next thing to look for in big science. The work didn't stop there. There were actually faculties who picked up the targets that we thought were important, our model said were important, and started generating essays on them. Because if you need to test an inhibitor, you need to first see if the function of the protein is getting inhibited or not. If the function of the protein has to be evaluated, there has to be something to assay. So there were teams who actually developed assays and identified potential inhibitors for some of the targets that were predicted by our model. So it was not just computational study, it was followed up appropriately with several experimental studies. So from a social network, we generated a biological network for MTB. So how people came together and got together all the knowledge on TB on a single platform. Now, identification of drug targets like the proteins that are acted upon by drugs was one part of the story, but then was an entire separate team working on identification of drug candidates themselves. And as you can see, I'll not describe it, but there were several studies which were done to identify potential drug candidates. One of the largest studies in that series was Molecular Property Diagnostic Suite, and Dr. Shastri must have already told you about this. So this was one of the largest exercises where we want to map the entire chemical space and to see if we can bring all the analysis that we have done on targets and on molecules together on a common platform. So consolidation of what was done so far. All of this was also followed by cloning of these proteins into E. coli. Now, for some of you who are not biology background, MTB is a BSL-3 pathogen. It means it requires a specific safety laboratory to be grown. There are very few labs in the world and in India which actually have the infrastructure to grow Mycobacterium tuberculosis. Now, if we want crowdsourcing to work, then we cannot limit ourselves to only work with people who have the right infrastructure, but we still want them to work on these targets. So what we did, we took MTB, we grew MTB, and we isolated its DNA. Now this isolated DNA was used to clone individual drug targets which are now non-infectious, and those cloned libraries can be distributed to anybody with no specific need of a BSL-3 facility. And this actually was done in collaboration with Shastra University, ANDC, and Miranda House, and for a simple reason that now we are moving from in silico, which was done on the web, to something that required physical infrastructure. So we actually utilized the existing infrastructure in colleges, and students who were doing very boring experiments from time and long, they actually were now doing a very different experiment of generating a resource that the world can actually use. Likewise, we also did an open chemistry initiative. Now, if a compound has to be tested, it needs to be synthesized. And who will synthesize that? So we actually again generated platforms where we got together chemists across the country to contribute these molecules and move forward in the drug discovery domain. Now, the collective effort led to generation of 10,000 compounds. It's not a small number. in like several hundred PhD students are and project staff is required to arrive at these numbers, and it is not easy for a single lab or a single group to achieve this. But when several people come together and several labs come together, they can collectively do it. And what this collective effort has led to is identification of 11 potential anti-TB candidates that were never reported before. So we made a new discovery after putting together so many people at the same time. Now, I have not been able to show you exactly and detailing every part of the structured collaboration, but this was an open, collaborative, distributed network, because right from all the labs that are marked here to colleges, to universities, to also hospitals, we also got a large number of private partners on board. We also collaborated with several international institutes and organizations. So it was like bringing the world together on a single platform to solve a problem that is so hard to treat. Now you understand that these problems are not easy. Look at the kind of expertise and the kind of infrastructure and the resources all of these people might have got on board. And still, TB is a problem. So the point is that these things are hard and we need to collectively work together to solve them. Now, as you can imagine, we could not have done this work over emails. So we required a platform that allows us to work in a virtual laboratory. So we partnered with Infosys and developed an indigenous platform that allows us to do this. At that time, there was no platform that allowed us to run a project of this magnitude in a seamless fashion. Imagine now you are on MS Teams, I'm giving a talk and you're listening to the talk. You can give Q&A and that's all. Now imagine I actually add other features to MS Teams. Say for example, there are ways in which you can, so I have a very interesting idea. Would anybody like to work on that? Somebody says, okay, I like the idea. I will work on this project and generate some results. On those results, somebody else are able to comment. And some of these things are either computational or non-computational. So you basically need all of this on the fly. And this is what this portal allows us to do, was to bring together the community onto a common platform and execute this project in a very seamless fashion. And this is basically to tell you that we utilized pre-existing resources wherever we could find them. So the entire network was built on NKN, the National Knowledge Network, which is like a gigabyte size internet bandwidth that we got. We collaborated with Infosys. We collaborated also with CLAB for the resources. So we always work with partners who were ready to move forward and help us in setting this network. Now, these are contributions from my lab over last 10 years. I've done several projects, almost all in a crowdsourcing fashion by engaging volunteer community, so that you actually, this is a way of training people on things that are normally not taught in colleges or university systems. And this is a way of us to understand it actually allows all of us to contribute. So I've worked with over 500 students. Like, I was very conservative here. I worked with actually over 1,000 students on several genomics and drug discovery projects, both wet lab and dry lab. And some of these students were school kids, 11th standard, 12th standard. Some of these students are from undergrad, post-graduation, PhDs, professors, lecturers, researchers, both from India and abroad. So it's a very, very heterogeneous mixed community that I have worked with over several years. And each one of them contributed the best they could. So that's what I call as individual competence to team delivery. There were so many people I worked with, and I could not place everybody's picture on the slide, and I'm really sorry for that. But there are so many people, and every time I work with anyone of I just want to thank everyone because they all taught me a lot. And we have together published in several journals over past 10 years. So it was not just a learning exercise. We learned, we worked together, and we contributed back to the scientific community so that the results and the observations can be taken forward. My favorite example of all of this exercise was this particular application. It is called DPAPS. So in this, we were the first persons in the world to generate a machine learning based model for predicting antibiofilm peptides. I really don't have time to explain what biofilms are, but for a general understanding, the way we stay in colonies, which allows us to have a sense of protection, microbes also like to stay in colonies. And when they stay in colonies, they actually share each other's armor, making them even harder to kill by antibiotics. So that is when we try to identify peptides that can disrupt these biofilms and to understand if there are certain properties of these peptides that we can learn and generate new peptides with antibiotic properties. Now, why this project is one of my favorite projects is because it was done by Pooja when she was in her 12th standard. She was just finishing her board exams when we started working on this project, and then subsequently, she went on to become the Cambridge Fellow, and now she is doing her PhD from York University. I have other examples from the people I have worked with from school and early undergrads who have done extremely well. And I think this is one way of identifying talent which is lying out there and to actually put them on the right track. So what have I learned from doing several Connect to Decode projects is, one, you need to have standard operating protocols. I'll tell you what it means. Since like now, we are not sitting across the table. But if all of us have to do one job, each one of us will have to have clarity on what that job is and how it has to be achieved. That clarity comes from standard operating protocols. What needs to be done and how. If we don't have that, each one of us will have a different understanding of what needs to be done and different ways of doing it. You can never put that together. Second, each project requires a different standard operating protocol. You cannot have one for all projects. So when we did the exercise on understanding MTB's interactome, right from undergrad students to professors were in the team. But when we were doing something like MPDS, it was heavily relied on people who are already research fellows because it was a little more skilled job than the others. So you need customized SOPs for each project. Of course, you need extensive training and discussion so that everybody is on the same page. If people in the team, like the online distributed team, are not communicating, the project will not succeed. So you need self-communicating communities. They need to start talking to each other. Very, very critical point, there has to be a roadmap of delivery and strict deadlines. If there is no clear direction of where you want to go, or there is no deadline as to when you want to reach there, none of these projects can ever be finished. And that is where you need collaborative platforms, not just for data curation, but also to follow up. And the most important learning is that always and always, for all of these things, be open to comments, criticism, and inputs. That is what helps you improve. Now, all of this learning, how we are applying in the current COVID era. So we actually have started a SARS-CoV-2 inhibitor like open repository. It's a rapid response team, I must say, because in a matter of 3 months, this team that you see here actually generated a very large repertoire of SARS-CoV-2 inhibitors at a single place and have done several analysis. I will not describe it too much. I would like you guys to go and check the page for Drug Discovery Hackathon. It has been posted as a challenge there, and we welcome you to join the challenge and be a part of this exercise. Now, as you can see that, and I've been telling you that, you need to think of always, keep learning and devising new things. So the most recent new thing that I have done is that last year when on my fellowship to France, I actually was able to generate an awareness drive, and this awareness drive has 2 components. It has a chatbot and it has a video game. So it's very unusual for scientists to develop video games, but that is what is the need of the hour is. If you really want to engage the community at the right age, you need to understand what really excites them. And I think every 12 to 16 or 17-year-old would like to play a game. And if while playing you learn, that's the best thing. This game is ready and we are planning to launch it sometime next month. So I'll keep you posted when the game is online. I'd like to take your comments. The game comes with a integrated chatbot because all of us have different knowledge on antibiotic resistance. So bot will understand how much you know and handhold you through the process because it is not possible for us to have 1,000 people sitting. So a bot will do that job. And this is very unique because I never came across a game, an education game which has a bot built into it. So what we did was pretty inventive and hopefully you get to see the game very, very soon. Now coming back to the final set of messages. The first message or the first word that I used in my today's talk title was believe. Okay. Now why did I do that? The reason I did that is because when we started the open source drug discovery project or when we were even thinking about doing something that big, if there is no belief that you can do it or this is something that needs to be done, you'll never do it because there is no precedence. So we were the first people in the world to actually create the largest open source community on drug discovery ever. So when you do something original, when you do something disruptive, you need to believe in it. And you not just have to believe in the cause, but you also have to believe in people. Somebody believed in me that I could do it. I may not thank him enough, but when Dr. Brahmachari actually took me on board for the OSDD project, he believed in me that I can do it. And since this work was never done by anybody else on earth, how would you identify people who can do it? There is no experience. So I think believing is a very, very critical component for you to do something original and disruptive. And I would recommend that you, and I think all of us do things which what we believe in. So believe in yourself and believe in what you do. I'll give you a very simple example of self-belief. I know there is a movie recently and that's why I'm giving an example. In Battle of Sarajevo, there were 21 soldiers who fought against an army of 10,000 people. This is nothing but self-belief. It's an unbelievable true story. A small defending force faced an overwhelming odd, and they won it. So this actually tells you about self-belief and the power of the mind. The second thing I wrote in my talk title is learn. Very, very critical. Learn new technology. Be like a Swiss knife, interdisciplinary. Every biologist today should learn how to code because if you don't, you will see yourself getting outdated pretty soon. Believe me, everything is getting automated. The data that is getting generated is outside any manual intervention. So if you don't start to learn to code, there are a lot of things that you can do with this data and in this data analysis that you are missing on. So be interdisciplinary. That is the key to the future. Because you are learning something that is not taught and you are interdisciplinary, you have to keep practicing. So you cannot always be taught everything, but if you want to learn something new, keep practicing all the time. And it definitely helps you learn better. And this is coming from my personal experience. When I wrote my PhD thesis for the first time, it was so poorly written that some of my friends told me that we don't even understand what you want to say here. It took me several revisions before I could write a proper thesis. But I took that as a challenge. And then I started writing and writing, and ultimately I end up writing 9 editorial choice articles for Science Translational Medicine, which no one has done before, at least from our country. So the point is something that I was not very good at when I practiced and improved. So the point is keep practicing and it will help you learn. Another message, when you are learning, never give up. I'm sure as kids, all of us have gone through the story of King Bruce and the spider. For those who don't know, Bruce was the king of Scotland. He lost several wars, and then one of the wars when he lost, he actually ran away and was hiding inside a cave. In the cave, he saw a spider trying to weave a web, but every time the spider was trying, it was falling down. and it couldn't basically go from one side of the wall to the other. And he thought that this spider cannot do this job. But the spider didn't give up. It kept trying. It kept trying and trying till the time the spider could actually spin the web. And that is when he thought that if a spider can do it, why not I? He went back, created an army again, and won the battle. So you should never give up. And that is, I think, a message that many people will give you. But I like this example particularly because he lost several times before he actually got up again and get going. Share. If you want to progress faster in life, learn to share. And the only way you can share effectively is by working in open, structured collaborations. And I'll tell you why. When you are open and you are sharing what you're doing, you can identify your failures early. People can help you do that. Structured is important because you need to be enjoying working in large teams and do reproducible science. Collaborations is because you need to complement your skill set with somebody else's skill set because nobody can learn everything. A few messages. It's just to show you that for open collaborations, what you need to be transparent. If you're not transparent, open collaborations don't work. In fact, collaborations don't work. It's important to learn from failures and not get depressed by them. You need to share failures with others so that they don't fail. Again, fail early. Explore new opportunities. Utilize as many people as you can and multiple stakeholders to see how do you arrive at solutions faster. And I think one of the major drivers of the projects that I do actually comes from a bigger cause, the emotional driver. If there is a problem and it is bothering me and I cannot do anything about it, I would like to work with people who can solve it. So all the projects that I have done so far actually dealt with understanding disease biology for new therapeutics. And I think a large student community actually, that attracts them because they know that there is an emotional component to it and they really want to contribute. So that's very, very important. You cannot, it's important to get connected to the bigger cause. Make collaborative science fun, healthy, and enjoyable by following certain rules. Now, people will say open collaboration and why do you need rules? You do need them. Reason is very simple. Even when we play childhood games like hide and seek or any other game, the first thing we'll set is who will do what will be the rules of the game, and then you start playing. Because if you don't have rules, you cannot even enjoy games, forget about enjoying doing science. So you need to have certain rules of the game so that everybody follows them and make collaborative science fun, healthy, enjoyable, and reproducible at the same time. What we discussed today was mostly drug discovery. But if I tell you, biomedical data today is growing at an unprecedented rate. It's heterogeneous, it is complex. It cannot be handled only by biologists. You need engineers, you need mathematicians, we need statisticians looking at it. And all of us know about the Large Hadron Collider in terms of one of the biggest projects that we have ever done, in terms of the data it generates. Go and search on the internet. Biomedical data is much bigger in size, much complex in heterogeneity. So we need challenges. So we need problem solvers who can address challenges like storage, analysis, sharing of this data. These are open challenges and nobody has clear solutions to this. Until the time all of us put our brain together in solving it, we are not arriving at a solution which is sustainable. And that is what I wanted to convey. It's not just drug discovery. It's actually the entire biological field is moving in a way that there is no way out if there is no open collaborative science for us to do. No matter how much technology we apply, I think common sense still is the baseline. And there is a reason I'm showing you these graphs. So if you look at the graph on your left, it actually shows you the number of people who drowned by falling into a pool correlates with films Nicolas Cage appeared in. There is a very strong correlation. But it doesn't make sense, right? The second graph is US spending on science-based and technology, and it correlates with suicides by hanging, strangulation, and suffocation. Again, a very strong correlation. Doesn't make sense. So statistics, technology, AI can give you many such things. But at the end of the day, the interpretation, the common sense, it was brings value in it. So AI and computing and common sense put together can actually give us solutions. Now this common sense or natural intelligence is collective in nature, crowdsourcing, when we can bring natural intelligence along with the other technology progress that we have made in terms of compute infrastructure or in terms of AI algorithms can really give us solutions. So we need to put the three together to arrive at something meaningful. So the take home message, and this is my last slide, is that all of us know that ideas needs wings to fly. But do you think an aircraft with only wings can fly? No, it needs a runway. So focus to become a reality, and the focus is your runway. You need to put energy in that idea to make it fly. But you always and many a times don't have a runway. Then you cannot fly a plane. That is where you need to innovate. You should be able to vertically ascend. You don't need a runway, you can still fly. So the message is, ideas are good, but you need to implement them with learning, practice and hard work, and keep thinking of disruptive ways of making that faster and more easily accessible. So this is basically acknowledging the people who I have worked with. So AB Open Lab, currently I have a lab here, and till last year I had a lab in France. So I had an opportunity to lead an international team of postdocs and students. I also actually am part of several other communities apart from OSTD, which is Joggle and Lectures Without Borders. So I actually very strongly want to again say I would like to thank all of the members here and several other people who are not here who have taught me and shown me the power of what believe, learn and share can bring to you. And with that, I would like to thank you and like to take up any questions you have.
Thank you so much, ma'am. I think those were wonderful messages and a wonderful lecture. And I think it is so relevant, especially at this time when the whole world is looking at the scientists and researchers to come up with drugs and vaccines against the SARS-CoV-2 virus. And you have explained so beautifully the various aspects of, drug discovery and the entire process that goes through to identify potential drug candidates. It was a pleasure hearing from you, ma'am, and I'm sure it was a learning experience for all the participants as well. As we can see from the comments that we are getting, somebody is saying that I'm so gifted to be attending this session. Well, somebody is saying that what a brilliant way to explain the points. So, like so on and so forth, so many comments we are getting, and that was really very wonderful, ma'am, and very informative. We do have some questions, so we're going to pick around 5 to 6 questions. And with your permission, I would like to pose these questions to you. But for this, I think I will request my friend, Ms. Monty, to conduct the Q&A session.
Sure.
Hello, ma'am. So the first question is from Abhishek Chatter. He would like to ask you about multi-target approach of inhibiting FAALS and FACLS as anti-TB drugs.
Right. So I'm glad somebody asked this question because this was one of the projects in OSTD. And this is important because these were two very critical enzymes in the fatty acid metabolism of Mycobacterium tuberculosis. The challenge is this: when we were actually looking at a single molecule to inhibit both the proteins at the same time. Okay. Now the challenge that we were facing is when we were optimizing the specificity of this drug, and now we know what specific means. For one protein, it was losing it for the other one. So the challenge is when you actually try to develop one drug for one target, it is hard enough, and you are trying to optimize it for two at the same time, it becomes even more challenging. So of course that's one way of going forward. But if you have ideas on how we can design compounds that can target multiple proteins at the same time in a rational way, we can try it out. On an average, I am not aware of any drug that works via a single target. If a drug is efficacious, it is definitely going and hitting multiple proteins. It's just that while we are designing it, it's hard, but naturally it happens. And it is specific to one target, but it does go and interact with the others as well. So it's not that it is specific to one, but rationally doing it to hit two proteins at the same time is extremely hard.
Thank you, ma'am. The next question is, the drugs used for treating TB kill M. tuberculosis, but it can kill other gut bacteria, which are essential for the body. So what about it?
Again, a very excellent question, and I don't know if you got a chance to look at my slides. When I was talking about elimination of the host proteins to gain specificity, we also eliminated all proteins that were in the oral or the gut microbiota to basically ensure that none of the proteins of the oral or gut microbiome are actually also have like the lack of specificity is not there. But the problem is that MTB is part of our natural flora. So it's not possible to design something which will absolutely be specific to TB. We can still have drug delivery mechanisms which we can optimize for that. But again, the point is that we eliminated proteins that were part of our gut microbiota when we were prioritizing drug targets. So we did that. If you look at the PLOS paper, it actually tells you how we actually achieved that goal.
Thank you so much, ma'am. The next question is from Ninat Patke. Does the mutant bacteria affect the gut microbiome?
A very intelligent question again. See, the point is pathogenesis is something that has to be understood in context. Like say, for example, when our microbiome is healthy, everything is fine. But when you have it's a concept called as microbial dysbiosis, which means that if these five organisms are staying in our microbiome and they are in some concentration and they are maintaining a healthy environment, now we either eat something or sleep poorly or take antibiotics. We do something that actually ensures that the concentration of this one goes down, right? And the other four remain there. Now this one which was not pathogenic in presence of this one, when this is not expressing, this becomes pathogenic. So the pathogenicity also is driven by the fact of the composition of the microbiome itself. So what you're asking is it's a very trick question because the pathogenic microbes are already there. It's just that they do not grow or outnumber the normal ones in a way that actually disturbs us. But they are already existing in the system. So we need to ensure that our microbial diversity is maintained, so that these pathogenic bacteria remain in check all the time. For a very simple reason, you know why we need the microbiome. So we cannot eliminate them, we need them, but we need them in an optimal dose. So that dose is maintained by other microbes in the microbiota.
Thank you, ma'am. Next question is from Akanksha Sharma. She wants to know how genetic diseases can be cured through drugs and how will it work?
Again, a good question. I think you should listen to some of the CRISPR talks. So right now, the state of the art is to do genome editing. So you can actually, if there is a genetic cause, which means that either a protein is missing or a protein is non-functional or a protein is overexpressing. There has to be something happening with the system which is not optimal, right? And right now with genome editing tools like CRISPR, you can actually eliminate those diseases. People have tried doing that earlier with stem cell therapy, with gene therapy, but there have always been challenges. CRISPR seems to be a very promising approach. A lot of people are working on it, but the challenge again is how do you know that when there is just one base to be changed in 3 billion bases, it will not go and do anything, which is again problem of specificity. It has to be so specific that if one mutation has to change, like say for example in sickle cell anemia, it actually should go make that base correct and should not do anything with the rest of it. A very hard specificity to achieve. But believe me, CRISPR is the future technology. And when I say learn new technology and be interdisciplinary, I was very tempted to write CRISPR. That is something that you all should read and learn about because it's a technology of the future. There is no doubt on that.
So the next question is from Anjali. Why vaccine resistance does not happen like that of microbial resistance? Ma'am, you have muted yourself.
I'm so sorry. Yeah, so when you say drugs and vaccines, you need to understand that they are developed with a completely different thought process. A drug is supposed to kill the pathogen. A vaccine is supposed to protect you. So a vaccine is basically again supposed to kill the pathogen by enhancing your immune system. So a vaccine is again a representation of a pathogenic strain so that your body recognizes it and generates memory cells against it, right? It's not that vaccine resistance does not happen. The problem is it is extremely hard to detect. And because it's the host which is evolving, and we don't have a generation time of 20 minutes, the evolution of vaccine resistance is also a little slower. But it is not that it does not happen, it happens. And that is why a lot of pathogens actually evolve and mutate to also show vaccine resistance, but it is still lower than what you see in drug resistance because here the dealing organism is the host and not the pathogen. And the host, given that we are exposed to so wide array of antigens that our memory cells can recognize a large number of variations. So probably the one message is harder to detect. Vaccine resistance is harder to detect. Second, our immune systems are exposed to so many antigens that probably there is already a circulating memory cell that can help us overcome even if there is a vaccine resistance. That is what I would, I don't think of a third reason for that.
Thank you so much, ma'am. Now I would request Elika ma'am to give the closing remarks.
Thank you, Ms. Monty. And thank you, ma'am, once again for so patiently answering all the queries and the questions. Our director, Dr. Sastry, he was really looking forward to be part of today's session, but he got busy with some meeting. So, but still, on behalf of him and on behalf of the SRTP team, I thank you once again. Thank you so much for sparing your valuable time to be with us this evening and to deliver this very wonderful lecture. Thank you so much, ma'am.
Thank you so much, and please do convey my regards and thanks to Dr. Shastri for having me here.
Sure, ma'am. Thank you. We would like to close this session now. Thank you so much, all the participants also, for being with us. Thank you.